Next speaker is Dr. Satya Arjunan from RIKEN. His title is Modeling Rod-Shaped Bacterial Division Site Placement Control with a New Spatio-Temporal Simulation Method.
Good afternoon. My name is Satya. I'm currently a postdoc at RIKEN with Koichi Takahashi-san's group. Today I'm going to talk about a new spatial simulation method that I developed during my PhD study at IOB. And how this spatial simulation method was implemented with the eCell version 3.1 and 3.2. So I use this spatial simulation method to model a rod-shaped bacterial division site placement control system. And today I'll present the basics of this spatial simulation method and some results of the modeling. Okay, first I'll describe the basis of E. coli cell division. And what are the requirements to computationally model the E. coli division? And after that, I'll describe the new spatial simulation method. And as usual, when you develop a modeling method, you have to validate it. So I'll give some results of the validation. And after that, I'll describe what are the problems with current division site control system of E. coli, which is consists of the mean D and mean E protein. And I'll describe a new mean E model which includes E-ring formation. E-ring is a kind of structure that forms on the membrane of E. coli. And finally, I provide some summary of my presentation today. Okay, in E. coli divides at the middle after about 20 minutes. So when it divides, in the middle, it can produce two equally distributed daughter cells. So E. coli divides in the middle with the help of FTSZ polymer. The FTSZ is a protein that binds to the membrane and forms a polymer. When it forms a polymer like this, it produces a force whereby it can constrict the cell. So with the constriction, the cell can divide into two daughter cells. So this is important because with equal distribution of the cell contents, only then both of the daughter cells can survive. Okay, there are also some possible mutations that can take place. If we inhibit FTSZ polymer formation, E. coli will tend to form a filamentous cell. That means it doesn't divide, it just elongates for a long time. So in the elongated cell, you can see that there are many chromosomes and there are no daughter cells formation. Okay, we can also, it's also possible to mutate the E. coli such that the FTSZ does not form right in the middle. So in this case, the FTSZ forms either at the pole of the cell or in the middle of the cell. So if it forms the pole of the cell, the polymer constricts at the pole and it forms a mini cell. So this mini cell doesn't contain any chromosomes. So therefore it's not viable. So this shows that the accurate formation of FTSZ polymer is important in order to produce viable daughter cells. So how is this FTSZ is made sure to be formed right in the middle of the cell? So there is this thing, this system called the mini-E system that controls the formation of the FTSZ polymerization. So over here you can see at 0 the t equals 0, this min C and min D GFP complex are localized at one pole of the cell. And then after about 20 seconds, you can see that they are localizing at the other end of the pole. And then at about 40 seconds, most of the localizations takes place at the one end of the pole, at the opposite of the pole. So this oscillation takes place at about every 60 seconds. Because of the oscillation, at the time when you average the concentration of the min C, min D, and GFP complex, you can see that the concentration is lowest at the middle of the cell. Because min C is an inhibitor of FTSE, this causes FTSE to only form in the middle of the cell. Okay, this graph shows the intensity of the GFP, and here this axis shows the position of the long axis of the cell. So when we average the time, you can see that min C is highest at the pole and lowest at the middle of the cell. Because min C inhibits FTSE, so FTSE can only form polymer at the middle of the cell. So it's important that this oscillation takes place every 1 minute in order to form correct division at the middle of the cell. So now, how does this min C system work in E. coli? So min C and min D, they can be either on the membrane or in the cytosol. In the cytosol of the membrane. So then they can bind to the membrane. After this, I'll explain the actual model. Then when they bind to the membrane, FTSE forms right in the middle of the cell. So this is the cartoon of the system. Okay, here's the pathway of the min DE system involved in the cell division control. So min D in the ATP form can bind either autonomously to the membrane. So because in the ATP form, min D conformation is changed such that it can bind autonomously to the membrane. You can also recruit min D already bound to the membrane, can also recruit cooperatively other min D from the cytosol to the membrane. So there's either one, so this K2 is much lower than K3. So cooperative binding is also very important in the system. And this min D, once bound to the membrane, you should take note that min C is always found bound with min D. So that's why I'm not including min C in this system. So min D, once bound to the membrane, it can also bind with min E from the cytosol. So once min E is bound to the min D, the ADP is hydrolyzed. And as a result, min D becomes an ADP form and the conformation changes. And min D is released to the cytosol again, and min E is also released. So this cycle continues as long as there's ATP in the cytosol. So with this kind of a system, it's possible that min D is found with just very simple model such as this. It's possible to produce oscillation in a computational model, which was published by Fong et al. So what are the things that we need in order? What is the motivation of my spatial simulation method? First, we need to understand what the basic method of cell division, such as protocells. The intuition alone is not adequate because sometimes by just looking at the GFP localization, it's not possible to uncover what are the basic mechanisms of the proteins association, dissociation, and localization. So we need to use quantitative modeling method to analyze and understand such systems, although they may appear to be simple.